All right, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, and 안녕하세요. And hello all. Welcome and thank you, SynBYSS people, for joining us the 185th seminar, and I call it a Technical University of Denmark Day. Happy New Year, everybody. Although it's already the 15th of January, I cannot believe it. Time flies. I embarked on my first trip this year on January 11. That will be a 17-day trip to attend two conferences and visit five universities to give talks and chair sessions. Now I am at the Microbiome Editing Godong Research Conference organized by a Nobel laureate, Jennifer Doudna and Dave Bickard, to chair a session and give a talk. It has been a fantastic meeting with great talks and discussions, meeting great old and new colleagues and trainees. I'm having fun. Actually, I went to bed again yesterday around 2:30 a.m. And then I wake up to chair this seminar and then missing the conference in the morning. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Jochen Förster. He is a professor at Technical University of Denmark and Chief Technology Officer, Novo Nordisk Foundation Center for Biosustainability. We met last year in Korea at a conference, and he kindly accepted my invitation to speak and kick off the 2026 SynBYSS seminar. And then we enjoy, of course, a lot in Korea. And then I really glad he basically starting this webinar series this year. Johan, thanks so much for your contribution to the scientific community and starting this year's webinar series with us, and the virtual podium is all yours now. And thank you again.
Okay, thank you, Teseo, for the introduction. Pleasure to be here and to present some of our new activities. Of course, I'm at the Technical University of Denmark, but actually I'm at a new institute which we have just started. Let me try to share a presentation. And what can you, oh, here it comes. And now I go into slideshow. I hope that works too. So, what? Oh, here we go.
Yep, working.
It works. Okay.
Yep.
Okay, I've got 5 to 10 minutes, so I will fly over the slides. I will not go very much into detail. But it's my pleasure to introduce a new center at the Technical University of Denmark, the Novo Nordisk Foundation Center for Biosustainability. We basically stopped it after 15 years. The funding ran out, but we have secured another pretty big 7-year grant from the Novo Nordisk Foundation to start a new initiative, which is referred to in short as BRIGHT. And it's the abbreviation for the Novo Nordisk Foundation Biotechnology Research Institute for the Green Transition. We've got a new director, Luc van der Wielen from Delft University, who's running the new center. So maybe very quickly, a background just to emphasize one thing. So I've been on and off, and you can read here yourself, on and off in academia, in large enterprises, and in startup companies, and also had the possibility to join and co-found one startup company down here, Fluxome, where we managed to get one product on the market, resveratrol, which now is with Lallemand in Canada or /Danstar in Denmark. So my major drive has been, and I think it's Aditya's drive too, is innovation and commercialization. So wherever I went to, the idea was to bring something to the market. Even at the university, the idea for me is to bring something to the market. So out of my PhD work could go into startup companies. At Flaxome, we developed a yeast process to produce resveratrol. Then maybe just to mention a few things at Carlsberg, total shift away from metabolic engineering, classical strain improvements, developing lager yeast, specialty yeast, non-alcoholic yeast to produce beverages with and without alcohol. And now since 1st of January, I'm continuing to be professor at Bright and am responsible for what we call the Bright Biofoundry at the center, a technology platform to help our research succeed in their missions. So it's all about towards a resilient bioeconomy, merging biotechnology, renewables, and societal innovations. When we wrote the application for Bright, our focus has particularly been on CO2, reducing CO2. Where do we want to do this? How can we contribute with biology to reducing the CO2? And it's difficult maybe to see on this slide. On the right-hand side, you have the green piece of cake. We're going big on agricultural applications. And on the orange cake on the bottom, you have a slice over slice the chemicals. We will continue to work on chemicals and materials. And our work will obviously be based on our achievements in the Novo Nordisk Foundation Center for Biosustainability. The vision is biotechnology as the potential to provide innovative, transformative, and scalable biosolutions. In Denmark, we like the term biosolutions over biomanufacturing, but it basically, at least for me, means the same. And we really want to bring back, or we would like to stay within the planetary boundaries with our solutions. The mission, I'll let you read yourself. We have 3 starting missions. They are referred to as sustainable materials, microbial foods, and microbes for net zero agriculture. Each mission is headed by a technical and a business lead. On the sustainable materials, we have people like Alex Tofgård Nielsen and Pablo Nickel. The microbial food program will be run by Morten Sommer, and the microbes for net zero agriculture will be run by Irina Borodina and Pablo Morales. This is a typical innovation pipeline. We love to have a lot of ideas. If we do a rough estimation at CFB, at the Center for Biosustainability, we are particularly working on technology readiness levels between 0 and 4, 5, maybe touched on 6. Bright will go, and we will really want to go higher on 7 and 8. We will work across DTU. We will work with colleagues from chemical engineering who are part of Bright, with colleagues from food, from electro, from construct, to really enable reaching the demo state. Strategy, goals, maybe just to emphasize, because it's short, I mentioned the interdisciplinary collaboration across DTU, which is actually set up. All the strategy discussions has been done with all the faculty at DTU. Another thing is we will have a strong focus on much more than we had before on societal needs. And then also we will establish satellites in Europe for the green transition. So quickly, Bright Biofoundry. The concept of biofoundry is typically automating strain design. We go beyond this. We really want to have a platform that goes from TRL 0 to 6. So we have on the left-hand side here the missions, which I've just introduced. On the right-hand side, you could say we have got the innovation. Center for Biosustainability started 40 companies in 15 years. We would like to stay as innovative, but we also would like to, in addition to starting own companies, to have more collaboration with large companies. The Bright Biofoundry could be considered as a mission enabler. We're a support function into the missions. We supply different things around the DBTL cycle, design, build, test, learn. And the whole thing is held by that we have researchers doing life cycle analysis, techno-economical assessment, absolute sustainability assessment, also with colleagues from DTU Sustain. We have a number of specialized teams organized. Let me start in the center. Typically, I forget this. The informatics platform run by Alberto Santos Delgado, a team of 15 to 20 people, really securing the metadata organization and the processing the data and get it ready for our researchers. We've got Patrick Van Nerf and Emre running modeling teams. We have an automation team dedicated to supporting strain construction, analytics, and adaptive laboratory evolution. We've got Vici, who is a lady who is doing the synthetic biology in the Biofoundry. Adam Feist has a group at Bright, as he had at the Center for Biosustainability on Ale. We have a pre-pilot plant where we can go to 100 liter producing samples, and we have the full repertoire for doing downstream processing and getting purified samples. And we have an analytics platform very much dedicated to metabolites, quantification, profiling, identification, and a little bit proteomics, not much. Otherwise, we do it in our neighbor department. And the last piece of cake, which I've jumped over, is a very interesting one. We've got an R&D kitchen run by a former chef of a Michelin star restaurant here in Copenhagen, who has a former chef from a 2-star Michelin restaurant in Hong Kong, and just another 1-star Michelin chef. They're working particularly in the Biofoundry together with the microbial food mission on turning food waste into food. Super quick, just to tell a little bit on how we intend to work. This is previous work from the Center for Biosustainability, but we have had a project for 18 months with the team of Morten Sommer and Leonie Jahn at that time, which is referred to as SynBYSS. I've promised to pay all the Biofoundry activities if Morten has a cool idea, and obviously a surprise he had, and he had to hand over a 100% project leader from his team, Veronica Ramos-Bellana. So we helped Morten. He had already lots of results to produce sweet proteins in yeast. We have a number of different sweet proteins, all in common. They have extreme high level of sweetness, typically 15 to 3,000 times higher than sucrose, meaning instead of using 1 gram, or let's say, 1 gram in a cup of sugar, you need to use 15 to 100 to 3,000 times less amount if you use the sweet protein. Has no calorie effect, better health effect, doesn't impact agriculture, etc. And normally, many of these sweeteners, sweet proteins, I should say, they are extracted from tropical plants. So what we did, we had strains, constructed new strains. We worked with the Ale team. There were growth defects. We did simple series transfers, all automated, not as manual as indicated. We have a set of 12 robots and liquid handlers where we can run parallel projects. We increased the growth rate by 60%. We went into our integrated fermentation platform, which today is part of the pre-pilot plant. We increased the titer to a pretty good titer that allowed us to produce samples in 30-liter fermentations, which we purified from the broth with IX, chromatography, and filtration, and some other tricks. Enough sample for 60 people to enter a sensory analysis and a panel whether the sweet protein matches the sweet proteins on the market, whether they're off flavors, etc., etc. And we got some very promising results. I apologize for not showing true data here. Morten has the idea sooner rather than later to put this into a startup company. Then on one slide, super short, because I think it's super cool, another project from the Biofoundry together with Tilman Weber, and he put Renata Siegrist in. And you can see here on the left-hand side that we used in a lot of different tools, anti-smash developed by Tilman Weber, etc., etc. We screened the 1,100 Streptomyces collection for BGCs, so basically almost up to 50,000 BGCs we were screening for fungicides, and we extracted BGCs from up to 100 kb and expressed those in a production strain. And then on the left-hand side, you can see we run bioassays, antifungals. We tried as a test case to find a novel antifungal against Fusarium. Fusarium is a pest on cereals like barley and wheat. It's hell for brewers to get barley with Fusarium contamination, and we're starting to see some candidates here, and we will further develop this technology. It's fully fully automated. It's fully half automated now. The extraction of the BGCs into the production strains. So we've had some success in having 22 Streptomyces strains, and we have had a number of different smaller inventions around this project. I'm running out of time. I wanted to show this. I think I will skip this. This is out of my Carlsberg time. If you're ever in the need for a non-GMO method, you hardly need it anymore. In Europe, you still need it, but still for microbes, this is a super cool way to use droplet PCR and identify a strain with any mutation you need. Have a look here. So I will jump over this. It has been developed at Carlsberg for plants initially, works super in brewer's yeast, and beverage and food industry is super, super, super conservative. They don't want to use GMO, at least in Europe and probably still some other spots around the globe. So big thank you to the BioFoundry team, obviously also to the researchers in the research projects, research groups from Morten Sommer and Tilman Weber, who all move now into mission projects. Acknowledgement to the Novo Nordisk Foundation. Pride has received 140 million euros for a 7-year period. And we would love to collaborate with you either on research projects, or we would also be happy to support you through our BioFoundry. Please feel free to reach out, jfor@dtu.dk. Thank you very much, Seuk, for the opportunity to present. I hand back to you.
Oh, fantastic. Oh my goodness. This is amazing. I really glad I hear a lot of my colleagues' name when you just present. But at the same time, this is exciting because this is really showing, synthetic biology or engineering biology really can deliver real product to the society, including delicious product. And this is what we need to do after, 25 or 6 years of synthetic biology or engineering biology. It's time to deliver something, real thing, in that way, basically benefit the society. So thank you so much. This is exciting. Last 4 or 5 days, I only slept 4 hours a day. You wake me up completely with
I
the
hope it lasts.
we do that. I
Thank
wanted to taste the
you.
one. Actually, I visit, Denmark. I actually saw even the kitchen to make real food. And then you mentioned Michelin star chef. Oh my God, I want to visit again, Denmark soon. Thank you so
Yeah,
much.
please pass by.
Yeah, absolutely. Yeah, I'm gonna do that. Okay, thank you.
So I stop sharing.
Sure.
Voila.
All right. Also, congratulations, the new star of the Bright. This is exciting time and exciting, the initiative, I guess.
Oh yes, yeah, it is. It is. We're really in. Let's say we're still on a honeymoon, in a honeymoon period, trying to get all things together.
Absolutely. Okay, so now the main speaker of today with a longer introduction, Dr. Aditya Sarnaik works at Arizona Center for Algae Technology and Innovation, in short, AJCATI. Arizona State University as an associate research professional. He completed his master's in biotechnology from University of Mumbai and earned his PhD from Institute of Chemical Technology, Mumbai, India. His research was focused on cyanobacterial engineering and their outdoor scale-up. Common theme today is, real, scale-up and commercialization, I guess. He joined Arizona State University as a postdoc in 2019, and where he worked on a number of projects within and outside ASU collaborations. He then joined Indian Institute of Technology Mumbai, one of the best universities in India, in 2021 as a research scientist. In 2023, he came back to the United States and joined Ajkati with Dr. Taylor Weiss in collaboration with a startup called HelioBioSys Inc. So again, the commonality is kind of real commercialization today. He's currently developing an end-to-end bioprocess at 1,000-liter scale for biomanufacturing of bio-based sunscreens from cyanobacteria. His research spanned outdoor culture scale-up, harvesting, biomolecule separation, identification, and purification with the goal of enabling industrial scale continuous production. He has a passion for teaching and hence he is seeking job opportunities in academia. With his skills in biomolecular and metabolic engineering and bioprocess development, he is keen on developing wholesale biosolutions for biomanufacturing. Aditya, thanks so much for your time today, and please take it away. All yours now.
Thank you, Tasyak. Let me share my screen. Can you see the screen?
Coming up. Yes. And slideshow probably.
Now?
Wonderful.
Yes. So thank you so much, Tasyak, for giving me this opportunity to present my work. So the title of the topic today is Scalable Biomanufacturing with Cyanobacteria. But before proceeding further, I would like to quickly take why it's not going by. Okay. So I'd like to briefly tell you about my focus areas of work till today, and then I'll jump onto my topic. So I work with protein engineering. I have experience with protein engineering, molecular tool development for metabolic engineering, wholesale biocatalysis, as well as in vitro reactive extraction and cyanobacteria engineering and bioprocess development. I'll be specifically talking about this particular part today. Within these focus areas, I work on designing one cell, two wells biorefineries for complete value addition. I'll talk about it in a while. Metabolic fingerprinting and flux analysis, biocell development and biomanufacturing. The core understanding of my work, I would say, is choosing the organism based on the bioproduction potential instead of over rewiring of a known engineering platform. That's why I have worked with E. coli, of course, Bacillus subtilis, C. glutamicum, and more than 13 different strains of cyanobacteria for metabolic engineering. However, for this time, I am restricting my talk to biomanufacturing using cyanobacteria, where I'm not engineering the stuff. So that is one interesting thing about today's talk. So with this, let me come down to this interesting article, which was published in Nature Communications in 2024. What is holding back cyanobacterial research? And they came up with these two interesting answers. One is large-scale production systems, and second is lack of knowledge. Now, I would like, so I work at Arizona Center for Allergy Technology and Innovation as CATI at Arizona State University. And we have, as you can see, there is a wonderful test bed facility maintained. Apart from these recipe ponds that you can see, we have flat panel bioreactors, we have tubular photobioreactors as well. But important thing we need to understand is even after more than 3 decades of research or intense research in cyanobacteria, engineering, we lack dedicated, mature technology framework for commercial scale-up of cyanobacteria. So this is one of the reasons that we do not have such kind of ample testbed facilities available. And another thing is lack of knowledge. Now, this lack of knowledge is for people like us who are working in this particular field since more than a decade. So, what do I mean by lack of knowledge is in the next slide. So, this is what has motivated me to get into this particular project. I tell you a quick story. So I was working at Institute of Chemical Technology, Mumbai, and we were engineering Picosynechococcus 7002, a marine strain of cyanobacteria for ethylene production. But there was a significant cost discrepancy between the product's market cost and the production cost. So how to bridge this particular gap? That was a big question mark. So we thought that we need to explore something from this organism which will add up value and bridge this gap. Apparently, at that time, our lab had got a project for separation of lutein and zeaxanthin. Now, interesting thing to understand is this lutein and zeaxanthin, both of them have similar structure except for the change of one double bond, and that makes them almost inseparable using usual techniques. So what happens is we have to use more number of downstream steps to get a pure zeaxanthin onto the market, and that's why I just checked yesterday. The cost of analytical grade pure zeaxanthin is around $800 per milligram. It's too much. So it's almost like a diamond in hand. So we thought that let's explore these carotenoids from cyanobacteria. And when we explored the pathway, we realized that cyanobacteria do not produce, or at least this particular Picosynechococcus does not have the pathway for lutein production. And it can produce lutein-free zeaxanthin, and that was a wonderful moment for us. So we made a title of One Cell, Two Wells biorefinery, where one cell is engineered for production of certain compounds and also explored for its natural bioproduction ability. We've filed one patent for recombinant zeaxanthin production from the system. So all in all, it looks somewhat like this, one cell, two wells biorefinery. And we tried to extend the same ideology to chlamydomonas, which we had got for a bisabol production. Now, ethylene pathway and carotenoid pathway are not a competing pathway, but even after having the competing pathways on hand, we saw that we are still getting a good carotenoid production from the system. So this was a wonderful thing. Now, what is a take-home message in this entire work is not explore carotenoids. The take-home message is these organisms have a huge potential for production of precious biochemicals. And this is a legacy of more than 3 billion years. When I say lack of knowledge is at this moment, many of us know that cyanobacteria fix carbon dioxide and get it into the biomass, but I would say they are the reason we exist today. Because they chose cooperation or conflicts, we exist today. So naturally, these organisms produce pigments, antioxidants, proteins, extracellular polysaccharides, and cell protective compounds. But apart from that, these organisms have been engineered and commercialized for various different kinds of products. My talk will be specifically restricted to these two categories of products today, the natural chemicals from cyanobacteria. With this is a kind of brief title of Biomanufacturing Natural Sunscreen Components from Cyanobacteria. As Jochen said, it is like biosolutions towards biomanufacturing, I would say, and the paper is in writing. So these are the points I'll be covering. So before starting again, I have to mention this man, Professor Franz Greiter. And I got to know about this person from one book chapter called The Man and His Work by Dr. Arbach from 1991. So we do not know about this person at all, but we know the product he has given us, and that is the sunscreens. So he made the first sunscreen, being a professional mountain climber, and that made him a successful businessman, I would say. So with this, we have a number of such sunscreens on market, and all these sunscreens have these chemical-based UV filters in them. And this is the usage frequency. This was one of the survey that was published in 2022 about that. Now, initially, we were considering sunscreens as the cosmetic products, but they are no more cosmetics, but they are the over-the-counter drugs. So that's wonderful. We have an established sunscreen market. But what is the disadvantage of overuse of these chemical-based UV filters? So overuse does not mean you are putting more and more sunscreen on your body. Overuse means bioaccumulation. So let's take this particular background understanding. So we put sunscreens on our body, we go to beach like Florida, Miami, everywhere. We take shower on the beach, and that water gets into the ocean. Now with this, all these chemically synthesized UV filters, they get into the ocean, and it causes significant issues to the aquatic ecosystem. Instead, if we have any bio-based UV filter, not a chemical UV filter. Now, all these organisms which are present in the ocean, they are naturally programmed to produce such kind of UV filters. So if I have any bio-based UV filters, it's like I am getting things from ocean and I'm getting releasing those back to ocean. So there is no problem of bioaccumulation per se. So mycosporin-like amino acids. Mycosporin-like amino acids are known to have a good UV protection ability. They work by the mechanism called as button on a string mechanism, that they absorb UV light and dissipate it as a heat and get back to its normal configuration again. So MAAs are produced by a number of organisms, fungi, including cyanobacteria. So for my current work, what we did is we used a cyanobacterial consortium, which was designed by Heliobiosis Incorporation, a startup under NSF, SBI and STTR grant. Rocco and David, they are the pillars of this incorporation, I would say, and they are doing a wonderful job. So with this background, let's go ahead. So the question is, if MAAs are good, if they are good for the ecosystem, are they better than the existing chemicals? Are they better than the existing UV filters on market? So that is a question. So I have asked 4 questions to myself, and we performed the experiments from that perspective. So the first question is, are MAAs better UV filters than existing chemical-based UV filters? So we performed a spectral analysis, and we saw that they are at par compared to the existing UV filters on market. So performance, wonderful. Second question is about durability. So if I'm using into a commercial formulation, can MAAs sustain under harsh dynamic natural light conditions? So we exposed it to Arizona sun, and we saw that under a little acidic condition, there is absolutely no change in their activity. So this was very interesting because a healthy skin has acidic pH. So this was a wonderful finding for us. So for light, yes, it works. Now the question is whether it can sustain our MAAs durable over extended time and high temperature. So we performed an experiment over 2 months, 60 days at room temperature around 22 degrees Celsius and 40 degrees Celsius using a normal phosphate buffer, 10% glycerol, which is usually used in most of the commercial sunscreens, and exopolysaccharide from the same consortium. And we saw that with exopolysaccharide, there is hardly any change in the absorbance activity. So that was a wonderful takeaway for us, that they are very much durable for temperature as well as light. And last question is, if I want a broader spectrum protection, can I use it with other sunscreen, and can we boost the UV absorbance of existing sunscreen formulation through these MA spiking? So we performed the similar kind of analysis. I am not specifically showing the names of the sunscreens, but we have sunscreen 1, 2, 3, and a non-sunscreen lotion, and I have put their compositions below this. So we spiked them with 0.05 millimoles of MAs. Now understand that even at almost 15 to 20 times lesser concentration, they are almost doubling the absorbance of the sunscreen formulation. So that was a wonderful finding for us. And with this, we can say that MAs can make, I would go a step ahead and say next generation sunscreens in this case. But the question is still how to manufacture them. So we targeted two products from the system. So one is mycosporin-like amino acids, which work as UV filters and also has antioxidant activity. And we have exopolysaccharide from the same system, which has anti-aging properties. So we get two reagents or two components of sunscreens from the same consortium. Later on, we also realized that these cyanobacteria produce most of the UVA absorbing sunscreens intracellularly and UVB absorbing MAs extracellularly. I'll talk about this in further slide as well. So we have two types of MAs, extracellular MAs and intracellular MAs. So now the question is about the cultivation. So I went from 1 liter to almost 1,000-liter scale. We can call it like a pre-pilot scale demonstration. And for this, we used, I'll recall, the HBS cyanobacterial consortium. Now, in this consortium, we have a nitrogen fixing cyanothece. What is the advantage of using nitrogen fixing cyanothece, as you do not have to add any extra nitrogen to the system. So that gets down the cost, and it is good for the scale-up and getting the final product cost down. Okay. So what we performed is we wanted to see the performance of these organisms in presence and absence of external nitrogen source. So we performed an analysis in flat panel reactors. In a greenhouse we have, as you can see, the UV light is hardly anything there, but we have dynamic natural light conditions existing over there. So if you see that with and without nitrogen, their performance is almost equivalent. And they consume nitrate almost within 10 days. So wherever you can see the drop, that is the place where we are replenishing the culture for continuous growth. And we also saw that their pigment concentration, chlorophyll and carotenoids, it is also not significantly affected with and without nitrogen. Now the question is about the production. So we targeted UVA and UVB absorbing MAs. Now, as I mentioned, this is extracellular plot, and this is for intracellular MAs plot. Extracellularly, you have higher amount of UVB absorbing MAs as compared to UVA absorbing MAs. And intracellularly, they are almost equivalent. Why? Because there is no UV light because of which they will be induced. So we are producing on an average 15 milligrams per liter of MAs intracellularly. And we are producing around 8 to 10 grams per liter of EPS, exopolysaccharide. Now, I have not mentioned it in the further slide, but we can actually go up to 20 grams per liter of EPS, exopolysaccharide, over the period of time, and which is one of the, I would say it is one of the competitive titers on market. So EPS is one of the high rated product along with, excuse me, along with MAs in this particular case. Now this was at 110 liter scale. Now we raised it into an LGM tubular photobioreactor at around 800 liter scale, where we had some UV light. Now this is also not a significant amount of UV light, but some UV light outside, and you can see that the UV, the intracellular production is significantly higher as compared to extracellular MA production. So we could reach almost 30 milligrams per liter of MAs intracellularly, which corresponds to around 5 grams of MAs per kilogram. But again, this particular ratio is not commercially significant ratio, I would say. So this was one of the interesting findings. Again, we could reach a good EPS production. Now, I would like to highlight a small portion over here, that when you see that the exopolysaccharide is almost consistent throughout the duration of cultivation, where I say there is a drop in the amount of nitrate or the sugars, I see that there is increase in the exopolysaccharide production. What do I mean by that? Is whenever the cells are happy and performing their normal metabolism throughout the cultivation period, they are not producing more amount of exopolysaccharide. But as one of the components in the medium goes down, they cannot direct the fixed carbon towards the biomass. So they accumulate that fixed carbon towards EPS. So whenever cells are stressed, we can actually produce higher amount of EPS. With this, we thought that if we have to exceed further with MAs as well as EPS, we can give some artificial UV induction in this particular case, where cells will improve the amount of MAs, and because of the UV stress, they will also increase the amount of some exopolysaccharide in this case. So this was the takeaway message. So with this, can we boost the production? That was the question. So I performed a simple experiment. I took my culture from the indoor flat panels, and I just exposed it to sunlight for 1 hour. As you can see, the culture has become very thick, and you can see this scum-like layer on the top of this. This is because of this EPS production, significant amount of EPS production, and it also has higher amount of MAs in it. So I thought of comparing the 12 liters indoor flat panel with 1000 liter outdoor pond, and we saw that there is almost 10-fold higher production rate of EPS as well as MAs. So this was something very motivating or inspiring for us, considering the further scale up in this case. Now the question was, this was more about the scale up perspective. Now the science is also very important. So the next question was MA characterization. So as I showed earlier, that we have two types of MAs. One is extracellular MAs and intracellular MAs. Intracellularly, we have more UVA absorbing MAs, and extracellularly, we have more UVB absorbing MAs. Now, I would like to mention here that cyanobacteria are naturally programmed to differentially distribute these MAs. What I mean, or what I hypothesize, that because of differential penetration of UVA and UVB light, they have more UVA absorbing MAs intracellularly, while more UVB absorbing MAs extracellularly because of their differential penetration. So we performed LCMS analysis of these MAs. So intracellularly, as I said, that we have more UVA absorbing. So I performed an HPLC analysis with that, where we saw that at 334 nanometers, we have higher absorbance, followed by 310, followed by 300. That means as we are moving from UVA range to UVB range, the absorbance goes down. That means we have more UVA absorbing MAs intracellular. So the prominent ones which we know they are definitely existing intracellularly are chinorin, mycosporin, methylamine, threonine, palatin threonine, asterina 330, and porphyra 334. Now, the main question was about the extracellular MAs. Now, in extracellular MAs, the trend exactly reversed. Now, in this case, we had 334 followed by 310 followed by 300. In extracellular, the trend was 300 and 310 are almost nearby each other, and then 334 is down. So we have more of UVB absorbing MAs followed by UVA absorbing MAs. And we could definitely found out chinorin and some of hexose bound palatin threonine outside. But apart from that, most of the MAs which were extracellular were bound to sugars. So we have to perform further analysis. So this particular study is still ongoing. I deciphered it can be some sugar derivatized mycosporin-like amino acid extracellularly. But confirmation is still on the way. So overall, this is the possible biosynthetic pathway. So this is the actual biosynthetic pathway for mycosporin-like amino acid. But I believe that this is the pathway for intracellular MAs, and this is the pathway for extracellular MAs for the consortium we have. Now with this, we still have some work in progress. So as I said, we are going at 1,000-liter scale. From 1,000-liter, we have to go to continuous 15,000 or more than 15,000-liter scale up with continuous sample processing, which is still ongoing. We are almost more than 50% in detailed MA characterization. We are almost 100% in biomass harvesting and separation, so we are good on that at 1,000-liter scale, and we are going to extend the same principle to 15,000-liter scale. For MA separation and purification, we are almost halfway. For EPS separation and purification, we are less than halfway, and we are still exploring on formulation and preclinical trials for that. For this particular work, we are in collaboration with multiple companies. So first of all, thanks for them. And based on this, as an independent researcher, I would like to say that as these organisms have their own mycosporin-like amino acid and EPS production ability, I want to take these species for biofertilizers production for regenerative agriculture. I'm thinking of engineering them for non-sugar carbon upcycling to produce bioplastic. Also for producing therapeutic peptides, because cyanobacteria are inherently programmed to produce higher amount of proteins. And of course, designing an integrated and continuous bioprocess for biomanufacturing. With this, I would like to thank my PI Taylor, our ASCATI director John, David and Rocco, of course, from Heliobiosis, our entire ASCATI team who works day in and out for the cultivation stuff. I would like to thank my previous advisor Arul, his lab, ASU core facility, Ryan specifically from Sandia National Labs, and my PhD advisors, Reena, Arvind, and all my collaborators. And special thanks to Taisiok for giving happy start to my year. So thank you so much.
Fantastic. Oh, this is amazing and exciting. So thank you so much. And also I remember I visited the ASCU and then I visited that open the pond and reactor. That was amazing. This large scale real product delivery possibility. That is amazing. So we see today all the way from automation, biofoundry, all the way to big scale reactor, and that is we need to achieve the as soon as possible and then continue to do so. So thank you so much. So let me ask, Johan has whether he has any question or comment, and then we start from there.
Yeah, no, thanks a lot for the tour, Aditya, the presentation. It's very exciting. So I cross fingers productivity will go up. Yeah, and the molecule itself is also really exciting, I think, and the purpose of producing is maybe the biggest thing here. And when that is said, I was wondering, and maybe I missed it. It's of course a natural produced molecule in many maybe mammals and fish and so on. But did you still do a tox test and compare it to what is available on the market in the conventional sunscreens?
Yeah, so we are in the process. We are talking to companies about the clinical testing, and it of course includes the toxicity testing as well. Yeah.
Then a more technical question. On one slide, I think you said you had, I think you have 5 gram of MAA in a kilo of biomass, I think.
Yeah.
And then you have 30 milligram per liter in the fermentation broth of MAA, I think. It was shown.
30 milligram is from the intracellular one, not the broth.
Oh, it's only the intracellular.
Yeah.
And not the extracellular. No, okay. Because I was just wondering whether the actual biomass concentration is still quite low.
Yeah, because this is not the dry cell weight. This is the biomass paste.
Okay.
And let's say the kilogram of biomass. So it is a biomass paste that we used. I mean, if I dry it, then MAs will go off. So
yeah,
I usually don't go for that.
Yeah. Okay. And then on it's very curious on in Bright we will start to work more and more on reactor design, particularly for in our case maybe C1 gas fermentation and things like this. But I think there's a similar challenge here on reactor design. Can you comment a little bit on what type of reactor you prefer? Is it this tank you showed on one of the pictures? Is it more the tubular reactors? Is it something totally different? Because I guess the big thing is to get in sufficient light into the reactors.
Yeah. So I will not share, but we are currently talking to Elgin about it. So we are planning to use their system. So our further scale up will be done in Amsterdam. And there we are planning to design a reactor where there will be an artificial UV induction.
Yeah.
So instead of inducing UV from outside, we are planning to do it from inside.
Okay.
And that will be like timely induction. But again, the time frame for which we need to induce based on the culture density, how much should we induce? Because again, there will be a shading effect, and the thickness of the culture will matter, density of the culture will matter. So those things are still underway, but we are currently in a very miniaturized phase where we are trying this design at 100-liter scale in another Netherlands-based institute, and then we will scale it up with at around 15,000 liters.
Yeah. Then on the other side, back to the cyanobacteria. So did you do some modeling, like genome scale modeling, to see whether you could find additional non-obvious bottlenecks, maybe combined with transcription analysis or something else?
The interesting thing I would say is this is a consortium. So yeah, so but doing it for one organism will be a little difficult. But as I said, cyanothyce is a king in this, and the rest two organisms are trying to balance its growth like as it is fixing the nitrogen, they are making a kind of a closed, compact ecosystem within themselves. So they are actually stabilizing the entire productivity. So the question is correct regarding the modeling. I do not specifically have any expertise in that, but we will go. I don't know whether we call it a top-down approach. That will first see which all types of mycosporin-like amino acids exist in this case. And from there, we'll go back to what kind of pathways we can target. Because except for Senecococcus, rest organisms are non-model organisms. So there is no genome available for them.
Okay.
And at this moment, I think we are the only lab we have which have these organisms. There is no other lab.
Well, that's really cool. So you have a really nice scientific challenge. So you have a heterogeneity challenge on the process design. And so is the heterogeneity stable in the fermentation, or does it change over time?
So this particular study was done back in Sandia National Lab. I think it was from 2018, 2019. So before I joined this project. And they realized that over the period of time, as I said, the consortium remains stable. So that is maintained. But during the scale up at 1,000-liter scale, we haven't found any discrepancy with the heterogeneity, as you said, but we do not know once we artificially induce it with UV light, what will be the change. So that will be the next question, of course. So correct, till now, everything is going blessed by sun, I would say.
Yeah. No, thanks. It's great. Remember BII in Copenhagen.
I just wrote down the answers.
So, Johan, any other question or comment?
I'm fine. It was very nice to listen to. I'm far from cyanobacteria. It was nice to see something different.
Sure. Thank you so much for your comment and question. So let me ask one quick question I may miss, but you making product out of this, amazing bacteria, and then you will generate a lot of biomass. And did you mention what you're gonna do with the biomass after extracting whatever compound? That gonna be very useful product also that will make the cost down significantly.
So you actually tapped on my confidentiality. Okay.
Okay. So you don't need to say anything about that also.
Oh, no. I can surely say that. So basically, we are working on separation of this particular biomass. So we try to combine two things. So biomass separation and biomass lysis are two things. We realized that most of the cost in downstream processing in biomass lysis, because you have to take the biomass and we lose too much of biomass when it comes to lysis. So we are planning on using disc stack centrifuge, where it can separate the cells in one run, and in the second run they lyse the cells as well. So you don't have to worry about getting a separate process for. So we are actually combining these two processes together. So that is something we are doing for cell lysis.
Yeah, because I worked for company for many years before I start my PhD, and then so my job was from the flask culture bioreactor scale up all the way to commercial scale bioreactor, and also separation purification, getting FDA approval. So I know understand entire process. That's why I'm asking that question, because I know which one is cost significantly, and then which one we need to improve to make the things commercializable. So that's why I'm asking that question. So other than that, many of my question actually already question asked by the him. So I have no more question. This is exciting. I mean, this is exciting. And then I hope, also hope you succeed. One thing I can also offer is I'm the as you know I'm the center director, working with more than 40 PIs from 6 different countries. With also we have the 20 plus industry partners, the companies, including the LanzaTech, because that's a relevant company to you because you are working with the sunlight and CO2. So if you wanted to join my global center, there's no fee involved at this moment, your company, and then I'm happy to consider and then happy to add your company to my center.
That would be absolutely nice. Thank you so much, Tex. I'll talk to Heliobas's guys, and they'll be happy to talk to you about it.
Yeah, absolutely. Just send me email and then we'll set up the meeting and then we go from there. Okay, I see now 3 minutes after 11 Central Time of US. So let me close. Also, I see some, the comment note from the audience, and they really appreciate it, and they said that thank you so much to both of you. So thank you again, kicking off this important seminar series with us. All right, so thank you all for joining and staying today. We'll meet again on January 22nd, Thursday, the same time, the same Zoom link. We'll have the Professor Hyun-Nuk Kim at KAIST and also Dr. Jae-sung Han at Columbia University. Actually, I'm gonna chair this one in Korea, probably very inconvenient time with the Korean professor. Interestingly, what a coincidence. As usual, the follow informal chat will occur without recording. Please stay here if you are interested in chatting with us. I will promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thank you again. I stop recording. 1 second.